I'd like to welcome Haijuan from the China Meat Research Center. Welcome. She's going to tell us about tissue fabrication in 3D bioprinting for cultivated meat as well. Well, Huyan, go ahead, please.
Okay. Hi. Can you hear me clearly?
Yes.
Okay. Okay, I'll start my presentation here. So thanks for inviting me to have this presentation here. And I'm so happy to be a representative to share our latest research with all of you here. My today's topic is research on skeletal muscle. I am Haijuan. Four aspects. Let's move to the background. I guess most of us are very familiar with the cultivating meat. It is promising and
we cannot see your presentation.
Okay. Oh, sorry. Can you hear me now?
Yes.
Can you say it now?
Yeah, it's not in presentation mode yet.
Okay. How about now?
Yes, perfect.
Okay. Oh, sorry. Okay. That's my topic, and yeah, I'm Haijuan Hu. I'm from China Meat Research Center, and I will introduce my topic for these four aspects. Let's move to the first part, background. Most of us are very familiar with cultivated meat, and we all know it is a promising and environmental friendly way to produce the meat when compared with the traditional livestock farming. And this is a general production process for cultivating meat using cell factory or bioreactor. But the texture of cultivating meat using cell factory or bioreactor are too loose to have real chewing sensation and to obtain a hunk of meat. So we need to find other ways to solve this problem. The researchers are striving for solving this problem. Then they find the 3D bioprinting technology. It helps the final products with real chewing sensation and help people to obtain a hunk of meat. So it is applied in cultivated meat now. And more people will find the bioink is one of the key elements to the 3D bioprinting. And many materials were used for the bioink, like alginate-gelatin or the modified gelatin methacrylate, and silk fibroin. They all have their advantages, but some disadvantages also existed. How to apply those materials as bioink in cultivated meat, and also to maintain their advantages, but discard those disadvantages. That is the key problem in solving it. So that comes to our research. So for our research, I will give you a technology roadmap. Firstly is the seed cell extraction, then for the cell scale culture, then for the bioink preparation, 3D bioprinting, and tissue culture. So all the cells from our research were all obtained from the black pig skeletal muscle. We call it PMSCS. After check the purity of those cells, then we use it. Then comes to the preparation for our bioink. So we find the two combinations. The first one, we call it GS group. Then we use GelMA and silk fibroin together in this bioink for the curing method and for the biocompatibility. And then the other group is AG group. We combine sodium alginate and gelatin together in this group. Then we compare their properties. Firstly, we see the mechanical properties. As we all know, when we culture it in a medium, it is a very important property. Then we find due to there are two different cross-linking modes and also with different gel concentrations, they all influence the final mechanical properties of the materials. Since AG group, they face some difficulties like structural swelling, shrink on a crack. So GS group are more stable when compared with the AG group. So GS group were selected for the following experiments. Then for the biocompatibility, after 7 days culture, we find the cell death and the viability results showed GS2 had the highest total cell number and the living cell numbers. So GS2 was selected for the following experiment. Then we further to investigate the proliferation and differentiation of cells in GS2 scaffolds. Then we find the cells stretched and fused and differentiated in the GS group. Then we find a muscle block structure was built with different layers, and also the GelMA and fibroin second group showed good rheological properties and also mechanical properties. It is good for the cells to grow on it and also proliferated and differentiated. But for our further research, we find as GS2 group, they need to use photoinitiator in the curing process, but it is inedible. So it is not allowed to add in food process. So in our further direction, we need to develop edible bioinks, especially in the gelatin-based bioinks. And we need to also to enhance the stability and printability of the gelatin-based bioinks. So that comes to our second part of the research. So we aim to prepare edible gelatin-based bioinks, and we all need to improve the printability, stability, and mechanical strength of gelatin bioinks to avoid those drawbacks. And we also need to reduce animal source components and also need to maintain good biocompatibility of gelatin. So we find two materials to prepare this edible bioink preparation. The first one is pectin. It is from the citrus peel or the sugar beets. It is plant-sourced material, and it is a macromolecular heteropolysaccharide. We implied it here is to enhance the printability of the gelatin. And also it has a natural porous structure, which is good for the cells to be attached only. Then the second one is transglutaminase. It was chosen for its mild reaction conditions. As we mentioned before, the photoinitiator is inedible. So we need to find another one to cure this scaffold. So TG enzyme was chosen here. And also for these two materials, they are all edible. They are allowed to be added in food processing. So let's give you a brief introduction about our technology roadmap. As I mentioned before, in our previous research, we used GelMA as our raw materials. So the photoinitiator must be added in a curing method. Now we made some improvements. We choose gelatin as the raw material, but it has no poor printability. So we find the macromolecule pectin to be added in it to improve this printability of the gelatin. Then in the curing process, we use the TG enzyme to cross-link it mildly. Let's give you more details about these properties. Then for the edible, prepared edible bioink, and we find pectin enhanced the shear thinning behavior of the gelatin bioink from figure A. And also from figure B, we can find the storage modulus was larger than the loss modulus at 37 Celsius degree, which indicating a very typical elastic solid characteristic after the TG enzyme cross-linking treatment. That is what we wanted, to have good stability at this temperature. Then we choose to check the mechanical properties. Then it can be found here. When we compare the compressive stress strain curves of this gelatin packaging hybrid hydrogels, for the cross-linked and the uncross-linked samples, it can be found with the addition of the TG enzyme, the strain were improved from 0.2 to 0.6. So this is consistent with what we observed in our rheological properties results. Then let's move to the ISCCM analysis. It can be found for the edible bioink, for the untreated gelatin and pectin matrices, and for the enzyme-treated gelatin and pectin matrices. A cross-linked hydrogel structure was formed in the enzyme-treated matrices. And also, it displayed smaller and uniform pore sizes, and also with a tight work structure. It can be indicated it has more strong magnetic strength from the mechanical results and the rheological results. They are in agreement. Then we did check the biocompatibility. Then we can see the cells stretched and grew on the bioinks effectively. As we can see the cell proliferation and the cytoskeletal distribution on days 5 and 12. Then we also did the live dead staining of cells, and we also did the cell numbers counting. We find the cells proliferated well on bioinks on different days. That comes to our conclusion. The muscle block structure was built with different layers. Then we prepared edible gelatin-based bioink without photoinitiator, and the printability, stability, and mechanical strength were improved after cross-linking treatment, and the cells attached and proliferated efficiently on the JS2 and edible bioink. And then I want to have some time to introduce our team to all of you. Our institute is China Meat Research Center. We have 3 main research field here. The first one is the meat science related research. It concluded also the meat processing and engineering development. And then the other one is the food quality and safety testing. And the last one is the publication. So we received grant support from more than 1,000, and we have many granted patents and standards. And for our team, we are cultivated meat group. There are 4 main research directions in our group. That is seed cell, bioreactor, medium, and scaffold. And this is our papers, book, patents, and the software copyright. And finally is our team leader, Professor Shouwei Wang, and also our team. Those work were done by all of us here, and I want to thank to them. And that's all. Thank you for your attention.
Thank you very much. Are there any questions?
Thanks for the great talk. I have a question about the SEM pictures that you showed for the porosity of material. I was wondering, how did you prepare the samples? Because I don't know how much you are sure about what you see as a porous material as artifact of sample preparation, or it is indeed that porous, the material that you have?
You mean for the packaging?
Yes, you had another slide that with gelatin as well, so it showed like a, yeah, this one.
For this?
Yeah.
This slide.
Yeah, so may I ask, how did you prepare the samples for SEM pictures?
Yes, it is with the same scale.
Did you freeze dry the samples before?
Yes. We
okay.
freeze it, then we to cut it in to the slices, and then to say the ISCCM analysis.
Yeah. Okay. Thanks. I think I got the answer.
Yeah.
Thank you.
Yeah. Okay. Thank you.
Another question? So then I have a question. You tell that when you treat your scaffolds with the transglutaminase, you see an increase in strain and a decrease in porosity. Do you also see shrinkage of your sample?
No, because it is a type of hydrogel, so it is like we also check the swelling ratio, so it didn't
It
crack all
it stayed the same
starch.
volume. Okay. Well, thank you very much again then.
Yeah, thank you.